invited speaker, Antonio Procopio, was proposed by Natividad Herrera Cano. Antonio Procopio is full professor in organic chemistry at Università degli Studi Magna Graecia at Catanzaro, Italy. Also, he is member of the Life Sciences Board from the PhD program at that university. He has authored more than high-impact 140 publications, book chapters, and patents. He supervised over 50 degree theses and 15 PhD students. His research interests are focused on green chemistry, food safety and quality, chemical analysis of biological samples, and synthesis of radiopharmaceuticals. In this line, today, Professor Procopio will talk about semisynthesis as a tool for broadening the health application of bioactive olive secoiridoids. Welcome, Professor Procopio. The screen is all yours.
Welcome, everyone, and many thanks to our chairman and the organizing committee for giving me the opportunity to present the research that we do in the deep south of Italy, precisely in the city of Catanzaro, where in its countryside was founded in 1998 the University Campus Magna Graecia, now named Salvatore Venuta Campus, from its beloved founder who passed away prematurely in 2008. The campus is far only about 7 kilometers from Ionio Sea, which is part of the Mediterranean Sea, so that the interest in the Mediterranean diet has sprung up spontaneously among our research activities. Surely many of you have already heard about the positive impact of the Mediterranean diet on human health, summarized here very succinctly in low incidence of cardiovascular disease and tumors. The traditional Mediterranean diet provides a diet rich in natural antioxidants and vitamins, in which the olive oil is the main source of fat. For long, the researchers identified, among others, the high content of unsaturated fatty acids in olive oil as one of the main positive factors of this diet. However, classical risk factors as serum cholesterol and blood pressure are almost the same for the populations of the Mediterranean basin and those of the countries of Northern and Eastern Europe. In fact, oleic acid is the predominant fat in the feeding of livestock such as poultry and pigs, so that its percentage in the Mediterranean diet is similar to that of other Western countries. The research thus turned its attention to the microcomponents of olive oil: vitamins, phytosterols, pigments, terpenic acid, flavonoids, squalene, and phenols, identifying some phenolic secoiridoid here in the slide responsible for various pharmacobiological activities such as antioxidant, antimicrobial, antiviral, and cytostatic activities. Phenols strongly contribute to characterize the flavor of extra virgin olive oils, giving it pungent and astringent tastes as a result of complex interactions between the minor constituents of the oil and the taste buds. Alongside the different sensory qualities that panel tests by expert testers have attributed to the different phenolic secoiridoids here reported in the table, many biological activities have been recognized to the phenolic secoiridoid of extra virgin olive oil, here summarized. For this reason, the European Food Safety Authority has expressed this scientific opinion by recognizing the following properties of this group of secoiridoid phenols from the olive tree, followed by a much more robust recognition expressed in the present health climate. A daily intake of 20 grams of extra virgin olive oil containing 5 milligrams of hydroxytyrosol and its derivatives such as oleuropein complex and tyrosol provide the expected beneficial effects. For about 20 years, our research group has been engaged in developing synthetic methodologies with low environmental impact that can respond well to the environmental sustainability requirements set by the 12 principles of green chemistry. Thus, looking around us, if you, for example, remember the sites surrounding our campus, it was quite natural that we started to think about the chance to use the developed methods to chemical handling of molecules coming from natural world. Since olive tree is probably the most spread vegetable species in the area, we considered the opportunity to apply our methodologies to the extraction and chemical manipulation of oleuropein and its metabolites, the major secoiridoid phenol present in olive leaves, drupes, and virgin olive oil. Thus, the health claim refers to olive oil polyphenols containing hydroxytyrosol and orotyrosol in their structure. All European and erythrocytes, the two main secoiridoid glycosides present in the olive fruit, must certainly be considered among them. During olive oil milling, the action of native beta-glucosidase rapidly transforms these two compounds into their glycol derivatives, which are then further transformed into several aldehydic and dialdehydic products, including the main derivatives oleasin and oleocanthal. Other phenolic compounds of olive fruits, such as verbascoside, nuzenide, and nuzenide oleoside, also contain tyrosol and hydroxytyrosol in their structure. However, these molecules are usually not recovered in the extracted oils, so all European erythrocytes and their derivatives are considered the only sources of these simple phenols in the oil. It was after Beauchamp and his co-workers published a study on the anti-inflammatory action of oleocanthal in 2005 that the chemical, biological, and nutritive properties of all these derivatives started being investigated globally. However, limited investigation on their biological properties have been carried out since the amount of secoiridoid phenols in extra virgin olive oil is too low to consider their extraction, while their total synthesis is too complicated to be sustainable on large scale. Thus, semi-synthesis is a smart strategy for achieving these bioactive molecules after a few synthetic steps, starting from a less important but more available natural analogues. In this context, in recent years, our research activity has focused on the setup of sustainable semi-synthetic strategies to transform oleuropein in its metabolites, oleasin, oleuropein glycon, and dimethyl-oleuropein. Oleuropein is the major secoiridoid present in olive leaves, which represent the main waste from the production of olive oil. Indeed, olive tree pruning produces olive leaves, which are usually scattered across fields or burned without any practical applications. Accordingly, oleuropein extraction from olive leaves has been considered as a way to value the olive oil production waste. The amount of oleuropein present in olive leaves depends on several factors, such as the cultivars, chemiophysical conditions of the soil, harvesting season, and pedoclimatic conditions. Moreover, the amount of oleuropein obtained by olive leaves extraction strictly depends on the storage conditions and extraction methods. We have set up a microwave extraction protocol to extract the natural ingredients from the leaves. Here you can see how in very short time it is possible to get a maximum extraction yield by using simple water for the procedure. Noteworthy same results can be obtained with a conventional heating system, but only by means of refluxing water methanol or ethanol mixture and prolonged extraction times. The biological and antioxidant properties of oleuropein and its natural derivatives have been extensively exploited in the pharmaceutical and food industry, and they've also been very popular as health-promoting products. Among them, oleuropein glycon, which is derived from the deglycosylation of oleuropein, has attracted considerable interest due to its biological and pharmacological properties, including antioxidant, anti-inflammatory, anti-cancer, anti-hyperglycemic, and beta-amyloid aggregation preventing activities. However, oleuropein glycon is not commercially available, and thus different chemical or enzymatic routes have been proposed for its synthesis. We proposed the semi-synthesis of oleuropein glycon starting from oleuropein using erbium trifluoride as a catalyst in wet acetonides at reflux temperature. These simple, selective, and eco-friendly semi-synthetic protocols lead to the glycon as a mixture of 4 tautomer forms, which was characterized by LCMS and NMR. The anti-inflammatory activities of the inextricable mixture of oleuropein glycon isomers have been investigated in vivo in mouse models of carrageen-induced pleurisy, in mice subjected to collagen-induced arthritis, in rheumatoid arthritis, and in the mouse model of spinal cord trauma. Taken together, the results of these studies show that olive oil compounds, such as oleuropein glycon, may be very useful in the therapy of inflammation. The hydroxy groups belonging to the hydroxytyrosol moiety responsible for the antioxidant effects of oleuropein, together with the hydroxy groups of glucose, are responsible for the high hydrophilic The hydroxyl groups belonging to the hydroxytyrosol moiety responsible for the antioxidant effects of oleuropein, together with the hydroxyl groups of glucose, are responsible for the high hydrophilicity of the whole molecule. This hydrophilicity limits the use of oleuropein as a natural antioxidant both in cellular, media and fatty foods. Accordingly, suitable modifications capable of protecting the hydroxyl groups without negative effects on the antioxidant activity can possibly produce lipophilic derivatives of oleuropein with improved bioavailability. Peracetylation has been reported as a transient chemical modification compatible with biological media, which can improve the drug-likeness of inactive principles by enhancing its cellular intake. Once inside the cells, endogenous esterases remove the modification, thus releasing the active principles. We realized the peracetylation of oleuropein with acetic anhydrides under the catalyst of erbium trifluoride using an eco-friendly and sustainable method reported in Part A in this scheme. Subsequently, we improved the method, planning a catalyst-free microwave-assisted version of this protocol in Part B in the same scheme. Both the methods we applied to prepare the acetyl derivatives of oleuropein and its natural derivatives oleuropein glycone and hydroxytyrosol, the experimental octanol water partition coefficients confirm the increase of lipophilicity of the acetylate molecules compared to the natural ones. The anti-tumor activity of these oleuropein peracetylate derivatives were evaluated against the human breast cancer cell line, and these compounds exerted higher anti-proliferative effects than oleuropein by an arrest of the cell cycle progression associated with a stronger antioxidant activity. Moreover, we evaluated the activities of oleuropein and its peracetylate derivatives against two thyroid tumor cell lines, demonstrating that oleuropein is able to reduce the proliferation of both thyroid cancer cells at micromolar concentration. The good results reported for the acetylate oleuropein and its metabolites encouraged us to consider the synthesis of oleuropein derivatives with modulable lipophilicity through the transacylation reaction of oleuropein and different fatty alcohols in the presence of erbium trifluoride as a Bain-Lewis acid catalyst. The entire proposed process accomplishes many green chemistry tasks for providing a new class of derivatives whose antioxidant activities were correlated with their augmented lipophilicity. As expected, all the derivatives were more lipophilic than oleuropein glycone. Additionally, the permeation profiles as well as the antioxidant activity test clearly showed that the increased lipophilicity is directly connected to the compound's augmented radical scavenging capacity, which allows us to infer the potential application of such compounds as additive in fatty foods and as protective agents against oxidative stress in cells. Hydroxytyrosol, with its three hydroxyl groups, presents the same problem of low lipophilicity. Also in this case, we set up a simple and efficient catalytic method to obtain acylated hydroxytyrosol derivatives with a long alkyl chain. C4, C18 saturated and unsaturated moieties have been conjugated to the hydroxytyrosol, and this new set of antioxidant molecules was evaluated as a function of their permeation profiles through the human stratum corneum and the viable epidermis membranes. The inclusion of a lipophilic chain in the hydroxytyrosol molecule enhanced its antioxidant capacity in the biological model investigated. The same hydroxytyrosol esters showed greater antioxidant activity than alpha-tocopherol and BHT to protect proteins and lipids against oxidation caused by peroxyl radicals in heparin homogeneity. As shown in this table, all the hydroxytyrosol fatty esters with more than 4 carbon atoms in their chain were considerably more lipophilic than a natural hydroxytyrosol. To test the topical application of the hydroxytyrosol fatty esters compared to the free hydroxytyrosol, an in vitro permeation study using France diffusion cells was performed on all the conjugates previously synthesized. The permeation profiles of the hydroxytyrosol conjugates show that different compounds increase the percentage of hydroxytyrosol permeated through the stratum corneum epidermis membranes. This conjugate offered the potential of being administered as a topical therapeutic agent for the treatment of cutaneous disease. As already said, a huge number of studies have appeared in literature related to the biological and pharmacological activities of olive European, the main secoiridoid of olive plant, and its molecular congener lyxoside. However, as a matter of fact, the endogenous beta-glucosidase liberated during the olive oil extraction process hydrolyzes both these secoiridoids, forming a series of degradation products, all of them less hydrophilic than original natural secoiridoids, and therefore more soluble in the oily matrix extracted from the droughts. Thus, extra virgin olive oil is scarce in the two natural secoiridoids, olive European and lyxoside, and much more abundant in their degradation products, a glycol forms of both, and their dielectric forms, oleasin and oleocanthal, besides hydroxytyrosol and tyrosol. For this reason, especially after Beauchamp and his co-workers published a study on the anti-inflammatory action of oleocanthal, numerous reports have been published about the biological properties of the phenolic secoiridoids of olive oil. However, little attention has been paid to oleasin in spite of its abundance and minimum structural differences with oleocanthal, so only limited information on its biological properties is available due to the many difficulties of isolating it as a pure compound in a sufficient amounts for extensive investigation. Erbium trifluoride as a low-toxicity catalyst. Nevertheless, considering the difficulty of extracting dimethyl from edible olives through a tedious and inefficient procedure, our research group tried to obtain dimethyl directly from oleuropein through the selective hydrolysis of the methyl ester group mediated by enzymes. The alpha-chymotrypsin bovine pancreas was selected among 23 hydrolyses for the oleuropein dimethylation step. Unfortunately, only 20% yield was obtained through this biocatalytic pathway, with hydroxytyrosol as the main product of the reaction. The slide shows the combined biocatalytic way to obtain oleasin starting from oleuropein. Finally, a couple of years ago, we proposed a more semi-synthetic, convenient way to obtain oleasin directly using oleuropein as a starting material via the alkoxycarbonylation. This reaction was performed by microwave activation of alkoxycarbonylation of oleuropein in simple water. We obtain oleasin with 48% yield in only 30 minutes, as pictured in the slide. The first step of the reaction liberates a beta-keto acid, which undergoes the second step, an easy decarboxylation, forming the oleasin as a final product. Among the various inorganic salts tested, sodium chloride can be considered the best catalyst of decarboxylation of oleuropein, considering also its low toxicity and large availability. Moreover, the complete disappearance of the starting material was observed, and the main reaction bioproduct, oleuropein glycol, represents itself an added value product. Since HPLC profile of the reaction mixture showed an interesting composition in oleuropein derivatives, we decided to use it without purification as additive for edible oils, natural, poor in antioxidant phenols. As we have seen in recent years, our research activity has focused on the setup of sustainable semi-synthetic strategies to transform oleuropein in its direct metabolites, such as oleasin, oleuropein glycol, and dimethyl oleuropein. However, still meager literature exists about analogous procedure for lyxoside and its derivatives. Lyxoside is purely represented in olive leaves. Nevertheless, the level of its main derivatives, namely lyxoside glycol and oleocanthal, are significant in extra virgin olive oil. All of them have been investigated for their anti-inflammatory, anti-proliferative, and protective activity against Alzheimer's disease. Since oleocanthal has attracted so considerable interest, several methods for its extraction and purification from extra virgin olive oil have been developed using both organic solvents and depurated solvents on both the lab and large scale. However, these extraction methods still suffer from serious disadvantages caused by the need for sophisticated instrumentation, elevated consumption of organic solvents, high process cost, and poor performance. Besides the extraction method, many strategies of total synthesis of oleocanthal have been formulated to obtain it in the largest quantities, but none of them have shown better results than 40% of yield in 10 synthetic steps. Thus, the first semi-synthetic alternative to oleocanthal was recently proposed by Sarikak et al., who, starting both from pure oleuropein and the olive leaves extract, passed through the enzymatic hydrolysis of dimethyl erythrocyte as intermediates, reaching oleocanthal with respectively 11 and 6% yield. The merit of the method was to try a ground-scale protocol to obtain oleocanthal, but the proposed pathway did not allow to obtain nor erythrocyte or its glycon. Now, we have just proposed the first green chemiocatalytic method to obtain the same semi-synthetic cascade, erythrocyte, glycon, and oleocanthal starting from oleuropein. Our retrospective analysis was addressed to obtain all the erythrocyte family derivatives from oleuropein as a starting material, imagining that after the hydrolysis of oleuropein to remove hydroxytyrosol and the paracetylation of the resulting intermediate, tyrosol could be esterified to obtain after the acetylation of erythrocyte. Once erythrocyte obtained, its deglycosylation and its CRAPCO decarboxylation will be able to lead to erythrocyte glycon and oleocanthal respectively. Mostly of the proposed synthetic steps composing our retrosynthetic pathways belongs to our experience, and they have been already optimized in terms of both efficiency and sustainability. Our efforts in this work we are mainly interested to find sustainable solutions for the remaining steps, path 2 and 3, especially for the esterification with tyrosol. The first synthetic step occurring was the basic hydrolysis, which was realized according to the literature by a basic treatment of oleuropein with NaOH. The hydrolysis mixture was used in the paracetylation step without any further purification. A solvent-free and catalyst-free microwave-assisted method for paracetylation has been used to protect glucose. The process required a long and difficult phase of refinement due to the instability of the acid. In fact, once oleuropein loses its phenolic part, it is very unstable even in conditions that are not too drastic. Subsequently, the protected acid has been esterified with tyrosol. We choose the Mitsunob's method, which proved to be effective with tyrosol without any preventive protection step on the aromatic OH. Nevertheless, an optimization study has been performed on this step to drive Mitsunob reaction conditions in a more sustainable direction. After several efforts, we selected the couple of Mitsunob's reagents, DCAD and the heterogeneous triphenylphosphine polymer supported in an ultrasound-assisted protocol, which afforded erythrocyte with 70% of isolated yield. Starting from erythrocyte, glycon, and oleocanthal, we obtained the following two sustainable protocols already published by our group. Actually, erythrocyte glycon was achieved with a yield of 61% by reacting erythrocyte in wet acetonizer in the presence of erbium triflate. On the other hand, microwave-assisted Craptor dealkoxycarboxylation in water in presence of sodium chloride leads to pure oleocanthal with a yield of 55%. In summary, we realized the chemiocatalytic semi-synthetic pathway floating to pure erythrocyte with an overall yield of 25% after 4 semi-synthetic steps, and to pure erythrocyte glycon and oleocanthal after 5 steps with an overall yield of 15 and 13% respectively. The reaction matrix and the geometric parameters of the trees corresponding to erythrocyte synthesis proposed give to an important improvement in the overall yield with respect to the method previously reported, with less reaction inputs, less reaction stages, and a higher mass effective process, thus confirming a global improvement in terms of sustainability of the present synthetic plan. All the methods here presented make available the following natural phenols from the olive tree: oleuropein, erythrocyte, and their derivatives. In recent years, we have sent these compounds around the world exploring their potential pharmacobiological effects, which are all connected to their strong antioxidant and anti-inflammatory activity. Very recently, we have focused on the possibility to use the individual phenolic compounds of olive tree as potential anti-inflammatory agents, demonstrating both in vitro and in vivo their effectiveness in modulating inflammatory parameters and reducing cytokine synthesis, which represent the most negative prognostic factor associated to COVID-19. Timely targeted therapeutic strategies based on drugs capable of blocking the cytokine cascade would certainly improve the clinical outcome of the disease. With the health emergency and the containment of the coronavirus diffusions requiring much shorter response times than those often needed for the approval of new drugs, the proposal of natural molecules active against the cytokine storm, which have already been used on the humans, may be a potential therapeutic option. So I want to greet you and thank you for your attention with this little spark of hope that solution to the pandemic triggered by nature may come from nature itself.
So thank you very much, Professor Procopio. Very nice topic. Now we have a few minutes for questions. Let me see here in the chat. We have a question from the audience. I will read some of it. From Natividad Herrera Cano. Thank you very much for the presentation. Very interesting, the dissertations. I would like to know which polymer was used in the Mitsunobi.
I need just a moment to check on the experimental part of the job, and I'll send it immediately on the chat. The name of polymer, I think, no?
Okay, okay.
And
yes.
we didn't try to make all European derivatives with the stearic and the linoleic acids. The long chain fat acids that we used to have the all European derivatives are reported in the lecture, in the seminar. And the name of the polymer that we used for the triphenylphosphine we'll put on the chat in the last minutes, in the following minutes.
Okay. Okay.
Thank you very much for your invitation. I would really like to stay in Argentina now, not in Italy. Also, if it's still summer here, a sunny. Yes.
We have another question if you want to answer. I can. Yes. Okay. From Juan Arguello, can we use less expensive Lewis acid like ferric bromide or aluminium chloride in the chemical modifications?
Yes, we try other cheap Lewis acid, but sometimes they are too toxic or they require a large amount of catalyst. Very often in not still in catalytic quantities, but you can arrive also to use stoichiometric quantities. So the catalyst name is not more appropriate for this kind of reaction. So you can save money, but you can't obtain the same results from the point of greenness of the method.
So, well, very thank you for the answer, and of course for sharing with us this meeting. Thank you again, Professor Procopio.
Thank you too. Bye.
Goodbye.